Let's talk about acid release. Now, acid release is going to occur in three phases. The first phase is known as the cephalic phase, and this is going to be happening with the sight, smell, thought of food, okay, or taste. The second phase is going to be known as the gastric phase, and the gastric phase is going to primarily occur with the chemical stimuli coming from food, as well as distension of the stomach. Now, when food enters the stomach, it is going to expand with the help of rugae that are found within the stomach. And once the stomach expands, then that is going to trigger the gastric phase, which we will talk about. And the third phase is going to be known as the intestinal phase. And this phase is going to start when the food enters the duodenum. Now, let's draw a parietal cell. And this parietal cell is going to have 3 receptors that are going to be important for these 3 phases. The first receptor that I'll draw is the M3 receptor. The second receptor that I will draw is the CCKB receptor, and the third and final receptor I will draw is known as the H2 receptor. Now, when we see food, smell food, taste food, or think of food, the vagus nerve will be stimulated, and the vagus nerve will release GRP as well as acetylcholine. Acetylcholine will bind the M3 receptor, and GRP will stimulate G cells to release gastrin. And then gastrin can act on CCKB receptors to stimulate it. It can also stimulate ECL cells. And ECL cells are important for the production of histamine. And histamine will stimulate H2 receptors. It is important to know that secondary messengers are going to result in the production of acid in the parietal cell. Now, M3 receptor and CCKB are going to use the second messenger system GQ, which is going to stimulate IP3 and calcium. And IP3 and calcium are going to then act on the hydrogen potassium pump. And when they act on it, then hydrogen is going to be released by the cell. Now, the H2 receptor is going to use the GS, which is going to release CAMP. And CAMP is going to then act on the sodium-potassium transporter for acid excretion. We also have chloride channels here, and chloride is going to then bind potassium. It can bind not potassium, but hydrogen that was released to form HCl. Now, what is important to know is that during the intestinal phase, we initially have acid excretion, but intestinal phase is going to be primarily responsible for inhibition of ECL, which is going to decrease histamine to reduce acid production. So our colon and ileum, they're going to release peptide YY which is then going to inhibit ECL and lower histamine production to decrease acid production. Now, we talked about hydrogen release. So HCl is going to be responsible for breaking down, or not breaking down, but denaturing proteins. They're not going to be breaking down proteins. So they're going to be denaturing proteins, and HCl is going to reduce acidity of the stomach. So HCl, once it reduces the acidity of stomach, it is going to convert pepsinogen into pepsin. And pepsin is going to be the enzyme that is going to be responsible for breaking down proteins into amino acids.